Hi, I'm guessing if you're watching this video, there were some technical difficulties. I'm sorry about that. If I could be in the room with you guys, I certainly would, but I'll do my best to pretend like I'm there anyway. So let's go to the first slide. And I just want to say it's great to be back. I'll also put myself down in the corner here. There we go. It's great to be back at the FTDNA conference. The admins here are so enthusiastic. You guys are a veritable army of volunteer scientists, so thank you as always. Most of you know me, I think. I'm Paul Mayer. I'm the population geneticist. The last time we had this conference in 2019, it was my first full year, and I just celebrated my 5th anniversary, so time flies. I've been absent since August, I think some of you know, as my daughter was just born, which is why I'm zooming from Tucson, Arizona. But I will try my best to pretend like I'm in the room, and I wish I could be there with you. My goal today is to give you guys a look under the hood. Some of you guys expect a technical talk, although I understand many of you want something more big picture. So, as always, I'll try to thread that needle. And I don't expect you to understand everything, but you'll walk away knowing a bit more what's behind the curtain. And by the way, that's a real Y chromosome in the top right there. They're actually shaped kind of like an X, but as every chromosome, they're not quite perfect, and they're what you call acrocentric, meaning they have tiny arms on one side, so they look a bit more like a Y. And when I was making this presentation, I realized that photo really reminds me of something. So anyway, I'm going to take my face away here. There are enough faces to look at. Um, I want to quickly brag about my team in R&D so you know who's behind all the work. We have Joran, our fearless leader, who's had his hand in pretty much every project and feature in some important way. Michael Sager, who is the living incarnation of the big Y tree. It's all in his head. He's also, fun fact, a fellow herpetologist like me. He's worked on crocodilian phylogenetics in the past. John is our newest member, and he's built the amazing Discover website. Miguel is our genetic anthropologist and expert from National Geographic. Roberta is the oldest member of the group. She was here pretty much since the beginning with Bennett, kind of the matriarch and the expert genetic genealogist and mitochondrialist extraordinaire. Ray's moved on, but he was a core member, our data scientist, and he's greatly missed. And Bennett is like an honorary member of R&D because he's always been and continues to be incredibly interested, invested, and knowledgeable. So with that, I'll give you just a quick background on myself. Like Connie, who's recently departed, sadly, and Michael Sager and others, I didn't start on humans. I studied the past, present, and future evolution of Yosemite toads in California for my PhD. The nice thing about working on toads is they usually don't care so much about GDPR or seem to care what I do with their data. A bit more difficult to find out their history, though, their ancestry. My core research areas are in evolutionary genomics, popgen, and phylogenetics and phylogeography, and of course on human evolution. And since I started in 2018, I've used some of my published toad work to build FTDNA tools, which I'll talk about shortly. This talk, of course, will mostly be about the last two topics, though, phylogenetics and human evolution. All right, so with that, there are three topics. I want to give you guys a quick crash course on human ancestry, how phylogenetics fits in, and the history of phylogenetic ideas. I think it's important for that context. And then I'll talk about our age estimates, how they work, and finally, I'll talk about our exciting new tool, Globetrekker. All right, so let's dive into the background. Today's talk is fairly broad and covers a lot of technical ground, but this is all in the service of the central question, who are we and where do we come from? We're very diverse, and the question is, how do we get that diverse? We are being buried alive under an avalanche of new PopGen papers every day. And, this is just the last month for my feed. So it's a good problem to have, but it keeps us very busy. And it can be difficult to keep up with. Just a few of the most recent discoveries are Otzi the Iceman had a lot of Neolithic farmer ancestry. We didn't know that. The discovery of the dragon man skull, you can see there, it's probably a Denisovan. And an interesting paper recently was about climate amelioration or improvement around 45,000 years ago, which is around the time of a major expansion in haplogroup K that led to human dispersal around Lake Baikal, which I'll talk about at the end. So we're constantly reminded of the astounding hidden diversity of human beings. To set the stage, remember we're all connected by a common genetic tree of sorts. Here you can see some of the extinct branches of that tree. Like at the very top right, you see the super archaic humans, and in the bottom, you see the basal Eurasians, which went extinct. And of course, not all of these branches are splitting. Some of them are coming back together. So the question is, where did our diversity come from? One possibility is that it's mainly from lineage splitting, like you see here, or perhaps by mixing it together. And to clarify, I mean mixing of our entire genome, not just the Y or the MT. Zooming in much closer, it becomes clear our history is dominated by mixing. We can see this in Native Americans on the left, in East Asians, and then in Europeans especially. We have probably the most sampling and information. So any population you can name, including the population of every person in this room, is a mixture of previous ancestral pops. Let's take an example. The Garifuna have a very interesting story. In about 1635, a slave ship carrying some Igbo, Nigerian slaves, wrecked off St. Vincent in the Lesser Antilles. And the Carib natives, who themselves had been earlier subsumed by the Taino people, those natives interbred with the Nigerians. But 160 years later, the British took command of the island and banished most of them to Central America, mostly Honduras, but also Belize and Nicaragua. They assumed the identity of the Garifuna, and even those who are now Americans can all communicate with their native language, which is related to Arawak. Really cool. And here's a chromosome painting of a Garifuna tester collected by one of our CSRs and anthropologists, Roberto, back in 2019. You can see the dual African and native ancestries there in orange and purple. So what population are they? Well, one answer is Garifuna themselves, a novel population. Another answer is a two-way mixture. But what about Native Americans? They too are a mixture from about 25,000 years ago. The ancestral North Eurasian people, represented by ancient DNA samples such as Malta boy in basal haplogroup R, those North Eurasians were neither West nor East Eurasian, but a now extinct northern group. So they mixed with ancestral Asian people to form Amerindians. And even West Africans are a mixture. There's a ghost population that probably gave us haplogroup A00 that mixed with ancestral West Africans. So our diversity comes from this process of splitting and mixing and splitting again, really. But what happens when we try to identify our ancestors using a DNA kit? Well, here's what happens when we use autosomal DNA. With some hard work and luck, you're mostly guaranteed to find ancestors stretching back 6 generations ago or so. But further than that, it becomes increasingly unlikely your ancestors gave you any DNA at all. Here you can see those ghost ancestors in red. They become more numerous further back in time. Let's compare this to your Y-DNA genealogy now. For females, of course, this could be your father's or brother's Y chromosome. Let's imagine you test your patrilineal cousin. This could be 4th cousin, 8th cousin, 16th cousin. And let's pretend that they know their genealogy. So that's their genealogy. At some point in history, your Y chromosomes share a most recent common ancestor. And of course, note that, due to what some people call pedigree collapse, others call inbreeding, there could be multiple paths to the same common ancestor. But let's just focus on that, one singular path that each of you has. That's the most recent common ancestor path. Now let's just ignore all the other lines in your family trees. I'm going to show you how much autosomal DNA is shared from your most recent common ancestor down to you and your cousin. You can see the shared DNA segments, shown in blue, get shorter and shorter as time passes. And I'm sorry if this gives anyone a seizure. Recombination shuffles the genetic deck between loci, increasing diversity in you, but decreasing DNA shared with your relatives. And by the way, this is why sex evolved. Basically to help us battle microbes, viruses, and bacteria, which mutate much more rapidly than people. Every sperm or egg cell has about 8 billion possible combinations or configurations, which makes us, unique little snowflakes. But if Y-DNA is different and it doesn't recombine, then the only thing that changes is that new mutations might appear each generation. And those mutations, shown here as asterisks, if they're shared by descendants, they can be used to construct a haplotree. And that is the premise of Big Y, right? So that's a quick and dirty intro about our ancestry and phylogenetics. We are thoroughly mixed human beings, not mixed up, just mixed. At least maybe I'm a little mixed up, but it depends on the day. And that goes for everyone in this room, or, your room and my room. Every population is a mixture of previous ones. Any tree that represents the totality of our ancestry would and could not be a phylogenetic one. Yet the non-recombining portions of our genome, particularly the Y, lend themselves to phylogenetic reconstruction. And since we're diving into phylogenetic topics, I think it's worth now giving a super brief historical context. What were the major discoveries and when? And by the way, this sculpture is Mark Wallinger's Y in Oxford. I thought it was a cool kind of diagram of phylogenetics to talk over. So first was Alfred Russel Wallace, and in 1855 he was the first to propose that species, or you can think of haplogroups, same idea, come into existence, coincident both in space and time, with their ancestors. So similar haplogroups are nearby each other because they descend from common ancestors nearby in time and space. And of course, geology and dispersal play into that. There's still Wallace's line recognized today. He accidentally found this imaginary line, this boundary between the two supercontinents of the past, the Laurasian and Gondwana continents from the Mesozoic. And on one side, it's really cool. You find one type of fauna, tigers, elephants, etc., Asian fauna. And on the other side, you find kangaroos, cockatoos, and so forth, all the Australian fauna. Darwin, feeling scared that Wallace might scoop him, published The Origin of Species in 1859. He basically invented the idea that we all take for granted today, that all organisms on life are united by a common tree. And that concept, based on overwhelming data, even back in 1859, was very quickly accepted. He also reinforced Wallace's ideas with many examples of his own. And for example, you can see here ratites, which are ostriches and moas, big birds, flightless birds, are geographically spread across the southern hemisphere and were shown to spread from one ancestor in the south. Well, a century passed, and long story short, there were violent debates, followed by reconciliation, followed by lots of math. And in the 1960s, a German scientist named Willy Hennig invented modern phylogenetics as we know and speak of it today. Before him, trees were not necessarily based on shared history, but rather on similar appearance, because in some cases that's all we really had. He invented the idea of parsimony in trees, branch-defining mutations, paragroups. Sound familiar? He also began to place trees on maps, and this is less known, and identified a few rules. For example, he thought the shape of the tree tells us the direction of migration. He called this the rule of progression, that movement happens from bigger glades to more nested ones, which is, a little more controversial now. Okay, and then the idea of the molecular clock was invented. The molecular clock. The idea was based on the observation that some proteins seem to change at a constant and predictable rate. Thus, a tree could be scaled from mutations into time. Since then, it became a more controversial idea and even acknowledged not to be true in most cases. For example, in vertebrates, which includes us. DNA and protein mutations are not constant throughout the evolution of vertebrates, but it still today offers us a nice simplification on which to build more advanced tools. Skipping ahead a little bit, the relaxed clock was first widely used in about 2006 when Alexei Drummond invented the BEAST software package. There are many reasons the strict clock might be violated. There are differences in human generation time, differences in population size, in basal metabolic rate, etc. Also, notably, the strict clock only works if DNA is not under any natural selection. And since the genomics era, it's been shown that almost all DNA, even non-functional, non-protein coding DNA, is under some selection. Okay, so that was the relaxed clock. Finally, beginning in the late 80s, two major leaps forward occurred in the field of phylogeography, or combining trees and maps. When John Avis first coined the term phylogeography in 1987, only mitochondrial or mtDNA partial sequences existed. Basically, simple networks or trees were constructed and then superimposed onto maps. It was a lot of circle drawing, circles, concentric circles, basically. Usually the goal was to find geographic barriers to dispersal, such as rivers, for example. Lacey Knowles brought this to a new level in the early 2000s when she started rigorously testing alternative hypotheses. For example, did species fragment into isolated refugia, or did they experience gene flow? And when exactly did these events happen? Thus, age estimation was brought together with phylogeography in the early 2000s in a very statistical, hypothesis-driven framework. Oftentimes, it wasn't just the Y or the MT, but genome-wide processes that were being modeled. All right, so now that we have a background, let's dive into age estimates of mankind, or times to most recent common ancestor, TMRCAs. This is the team that developed the age estimates of mankind. It's myself and Euron, who you already know, our friend Ian McDonald, who's a physicist and a U106 expert and expert on many things, who kindly contributed an important piece early on, and our friend Stefan Bettner, a statistician who has very enthusiastically been involved throughout. Let's quickly see what the goal of our method is, and then I'll walk you through the 3 phases of it. Bear with me, this part is slightly technical, but I'll try to keep it high level. All right, gotta get a sip of coffee to get through this part. So, the premise of our age estimates is that your genetic TMRCAs can infer your genealogical ones. Simple enough, right? Here's a tree that connects me to Michael Sager. We're both in haplogroup E, and our common ancestor lived about 3,200 years ago. Not too far in the past. The shape of the tree will capture some, but not all, of your patrilineal history. Of course, the tree will not resolve every father-son relationship because mutations don't happen every generation. And this is maybe the most important part. It is a purely genetic tool. It's independent of genealogies. It's entirely data-driven. Sometimes we get asked the question because, we get some estimates spot on. Do you or will you use genealogical info or ancient DNA? Sometimes people think we're using ancient DNA to calibrate our estimates. The answer is no and no. The benefit of this tool is that it's completely independent of the paper trail. It gives you an entirely separate estimate to use. All right, so what's involved? Well, notice that a tree does not automatically have a time element. See the jaggedness of this tree? Not all branches end at the same point. Most trees are units of mutations, and those are called phylograms. What we want is a chronogram, chrono meaning time, like this, where branches are scaled to units of years. All right, so a bit detailed here, but I think this is important to set up and gives you a context for everything else. So phase 1 of our method does the setup. We need to carefully choose our DNA, choose our mutations, choose our mutation rate, and our tree. Stable DNA regions are important to choose because we can't manually comb through everything. We use a combination of academic studies, Michael Sager, and father-son mismatches to vet our sites. And our 11.2 megabase region that we use is called FTBED. We don't currently use the XDR, but that is always a topic of future discussion, the pros and cons. Our mutation rate comes from David Posnick's 2013 paper and comes out to about 125 years per SNP at the median sample coverage. For our big Y tree, we're always working on the cutting edge, and sometimes we need to temporarily define branches before Sager even has time. So we place academic samples if we can confidently do so. We use high coverage samples only, of course. And, we place new branches wherever we can based on shared mutations and negative results. We also use a sort of scaffolding or backbone tree, which we call our calibration panel. It's constructed with about 100 big Y and whole genome results, sequenced to high coverage. And the question is, why do we need a calibration panel? Well, in the Y-DNA tree, there are these major haplogroups spanning from A, B, C, all the way to T, right? Many of these major clades have long stems, which are prone to estimation error. And also the tree is huge. So these clades make natural groupings, and this panel that we're using allows us to partition the analysis into smaller trees so we can work on bite-sized chunks, use a separate method for those long stems, and then compare the two methods for quality assurance and a sanity check at the end. All right, the next phase is complicated, so I'll just summarize very briefly as best I can. We merge Big Y 500 and 700 results, so their coverage is apples to apples. All SNPs have a minimum depth of 6 reads and quality of 14. The most important step is shown at the right. We must calculate the coverage. In other words, if 3 SNPs were observed on a branch, What was the maximum number of SNPs possible? It depends on which bases were sequenced by the relevant samples, right? Ian MacDonald contributed this part, which we call intersect coverage. Next is the meat and potatoes, estimating the TMRCAs. First, we convert every branch into time using both SNP and STR markers. We capture uncertainty using gamma distributions. Next, we use a relaxed clock method for calculating the initial TMRCAs. And finally, we resolve any paradoxes caused by the method and validate our results. How well did it work? Here's how SNPs are converted into time. For example, 8 SNPs divided by the product of mutation rate and the coverage gives us about 975 years for this branch. But after applying our gamma distribution, we see that the branch could be anywhere from 550 to 1750 years long, and that's the 95% confidence interval. So we get that uncertainty baked into every stem of the tree. We can also calculate branch lengths using STRs. They give us an independent estimate of branch length, which we can average together with the SNP-based ones. The purpose of this is regression to the mean, basically. Either SNPs or STRs might have extreme values. They could have very, very strange values. So averaging the two can moderate your result. We build a general additive model, a GAM, which relates GD back to TMRCA. And you can see that the curve on the right is a bit nonlinear, right? Okay, so how do we calculate these GDs for STRs? First, we only do it for younger branches, roughly 2,000 years older or more recent. The model uses high quality samples only. They have to have all 111 markers and the 500 plus markers. And we use a stepwise model. For multi-markers, which are a bit more complicated, we use what's called the genome addition, maybe addiction, the genome addition or subtraction model. And currently we don't weight markers by mutation rate, but this is something we may change over time. Another sip of coffee. All right, so one bonus of this STR model, it gives us the new FTDNA tip report, which most of you guys, I think, know about by now. So for example, here you can see the predicted TMRCAs for each GD level of Y111. Smaller GD, of course, has more certainty, more narrow distributions in time range. It's a very pretty rainbow plot. Also, certainty increases with the number of markers used, and this illustrates this beautifully. Here you can see the confidence intervals get tighter from Y12 to Y37, all the way up to big Y700. With Y12, the haplogroup could plausibly be from the Bronze Age, and with big Y STRs, we know the haplogroup is likely 400 to 700 years old. All right, so now we have a tree where every branch is scaled to years. So that's it, right? What's left? Well, notice the jaggedness of this tree. Branches don't all end in the present day, yet the same amount of time must have occurred from root to every tip. So this is not yet a time tree, and it needs further correction. There are 3 sources of statistical error we need to consider. I'm going to try my best to make this an easy metaphor. So the first source of error we call stochasticity. Each tick of the molecular clock is irregular. It's uneven. Think of your mailman. If he's like mine, then sometimes he comes at noon, sometimes at 3 p.m., sometimes not until 6. Occasionally he might come in the morning if you're lucky. His rate of mail delivery is constant. It's once a day, but each tick of the clock is irregular, right? So thanks to our sloppy mailman, each haplogroup gets its own squish factor or distribution. A second source of error, we don't know the mutation rate with perfect certainty, right? The Posnick rate has some error bars around it. So back to the analogy, maybe our mailman is partially retired and in actuality, he delivers the mail once a week, not once a day. That's a much slower rate. So all haplogroups get extra padding due to this uncertainty. Finally, as I alluded to earlier, a strict molecular clock doesn't make much sense to begin with, because different branches potentially mutate at different rates. Maybe our poor mailman noticed the price of gas and milk and saw the news about the war and has resumed once-a-day delivery to pay his bills. This is what we call heterotachy, or difference of rates across branches. Here's a cartoon of how relaxed clock smoothing works. The blue branch has a mutation rate increase, the red one has slowed down. These branches need to be normalized, and we'll see in a minute how. So to recap, there are three sources of error that require us to smooth out a time tree: stochasticity in the clock, uncertainty in the clock rate, and changes in the clock itself across the tree. The relaxed clock method we employ tries to minimize errors on every branch simultaneously. Longer branches are penalized the most because they tend to reflect the biggest rate changes. The branch errors are normally distributed, and in a weighted least squares regression model, we can model those normally distributed errors. We solve the model with either maximum likelihood or QPD methods. So the important part is that we're penalizing longer branches. So we go from this to this. But what if we've accidentally made a haplogroup older than its parent? This is a time paradox and might require some funny business, like playing Chuck Berry at some music school dance. No, I'm just kidding. If we have paradoxes, we simply add some time, some buffer time between those branches. We use 30 years, or about one generation if possible. If we squish down a paradox clade, then we also have to scale its downstream clades by the same ratio, right? And then we double check that no haplogroups are younger than someone's age, for example, 64 years. And finally, we have a finished time tree. Then we use customer data to validate its accuracy. Here you can see 384 pairs of kits compared to the estimated TMRCA compared to the actual one. And if you look at the tiny, it might be hard to see, but if you look at the only points that are not blue or purple, but they're actually red, those are ones outside the 95% confidence interval of accuracy. And what this shows is that fewer than 5% of data points are outside the true 95% confidence interval, which is very, very good. That's what's expected. We also use ancient DNA to validate our results. Not every ancient DNA sample is placed with full resolution on the tree. Hence, all we know is that samples shouldn't be older than their estimated branch. Anything up there to the left of the diagonal or overlapping the diagonal is good in this plot. So all looks good. Here you can see Roberta's genealogy, the Estes genealogy, used for validation. Different colors represent different major family groups. And remember, this is men only. 4 ancestors actually have defining SNPs, shown by the blue ovals. This is what you see on the haplotree as well. Here they are in the block time tree format. And the y-axis is time, of course. Countries of origin are shown on the tree tips and on the map. You can see the heat map of where the countries that have these haplogroups are. And all age estimates here are accurate to Roberta's genealogy, which is great. Just for comparison, here's the block tree before any time correction. You see that some branches have wildly different lengths. Here's the result for the famous Scottish knight, Sir John Stuart of Bonkle. If you assume a strict clock and sum up all tester descendants from Stuart, you'd get 1500 CE. Now this is clearly centuries after Stuart was actually born, which was about 1246 CE. The problem is his ancestral haplogroup, RL745, has a long stem, thousands of years long. So long story short, that stem sped up in the mutational rate from over 120 years per SNP to less than 100 years per SNP, pushing his haplogroup into recent times. The relaxed clock effectively removed 683 years from his stem, and now his TMRCA is spot on. And here's that beautiful corrected time tree. You can see the estimate is spot on. Very cool. Another famous example is the Jewish Cohen lineage, representing a priestly caste from during the First Temple period, or from 1200 to about 586 BCE. Sure enough, the clade coalesces about 650 BCE, near the end of the First Temple period before its fall, so this makes good sense. Using the age estimates in ancient DNA, Miguel then made our original migration map with about 100 major clades. Here you can see haplogroup R forming in Central Asia before R1b invades Europe in the Bronze Age. We next wanted to extend this migration map to the entire big Y tree. So that brings us to the final topic, Globetrekker. Here's the core Globetrekker team. We have Joran, Miguel, John, and myself. And as always, a billion other people helps give us input, and we're thankful for everyone who helped. And of course, as always, Michael Sager is building the entire tree that feeds into this. Let's just start by appreciating the beauty of the finished product first. These are Globetrekker migrations for Amerindian haplogroup QM902 and all its descendants. Everything you see, from migration paths to every aspect of the map, we created from scratch. Another cool view of this is to overlay every single migration path in humanity, migrational spaghetti. The colors are, of course, the major haplogroups A through T. So how do we go about this? That's what I'm going to briefly explain. There are many initial steps to filter and curate our customers' geographic data and publicly available environmental data. First, we must know the extent of continental land masses through time, because they weren't always as they are today. Global sea level was lower during the last ice age, creating land bridges such as Beringia, Doggerland, Sundaland, and the entire Sahul continent, shown as the white areas there. So all these land bridges existed in the past. Here are those sea levels shown backwards through time across human-wide DNA history. Notice that minimum sea level was about 139 meters below modern level, which was 24,000 years in the past. Land bridges existed at various times in human prehistory and then disappeared. So the British Isles were connected with continental Europe in a landmass called Doggerland. But you can see that that land bridge disappeared before around 6,500 years ago. A similar process is applied to glacial boundaries, which also provided or inhibited migration corridors. Here's Europe's ice sheet at the peak of last ice age. And here's the ice corridor that opened up for the Amerindian expansion. You can see that the sea ice boundaries, you can see ice boundaries at 20, 15, and 10,000 years ago. So between about 15 and 20,000 years ago, there was a corridor that opened up insularly. And of course, there was also a corridor on the coast on the Pacific side. Not all human migration occurred entirely overland. Some cultural movements, such as the Viking raids and the Austronesian expansion, used seafaring routes, right? When such routes were mostly coastal, we assume they followed as closely to existing land as possible. Coastal routes, those within about 200 kilometers of land, are thus simulated to prefer a path that minimizes offshore distance. Oceanic routes that are farther seaward may be subject to maritime conditions, so we use a special layer of oceanic current speeds and directions to simulate the most likely path of least resistance. Afterward, we manually curate these paths and decide, should we keep or toss it out? Because sometimes we have anthropological or historical information that supersedes it, and so we may want to just show a straight line across the sea if we don't like what we find. So there's some curation. Okay, so Globetrekker is entirely driven by customer data. This is a strength because it can overturn preconceived ideas, and that's always good. But we must carefully filter data. First, we filter out samples with country and haplogroup combinations that don't make sense for pre-Columbian travel. For example, Eurasian haplogroup R1b should not be in the US, nor should Native American haplogroup QM3 be in Europe before the Columbian era. Next, we conservatively filter out earliest known ancestor, or EKA, coordinates that differ from the listed country. Customers may accidentally provide conflicting geographic information, which is a convenient sanity check for us. We do allow a kind of liberal 500-kilometer buffer between the two locations to be lenient, and we make exceptions where possible, such as an R1b person who lists Germany and the US. Finally, we check each branch for continental outliers. For example, if a branch contains 4 samples, with 3 in Europe, but the 4th is in South Africa, we rely on the first 3. After the samples are filtered, we also filter the tree itself. We want to carefully ensure that single branches don't unduly influence the migration paths. If any branch contains only a single geographically informative sample, or samples are no closer than 2,000 kilometers apart, we collapse that branch. After preparing the environmental data and heavily filtering user input data, we begin the monumental task of drawing 50,000 lineages accurately onto a map. Remember that about 2,000 samples are ancient DNA, and those ones are particularly informative about the past, right? They're closer to the past haplogroups. Remember also that the migration map I showed earlier is Miguel's work. He used anthropological information to manually place about 100 branches, and this gives us a starting point. The other 50,000 are automated. Our main assumption in Globetrekker is parent haplogroups are centrally located between their downstream branches. All things equal, people should spread out at similar speeds, right? However, it's essential to only consider direct downstream branches to avoid sampling bias. So take this example on the screen. The majority of samples for this clade, about 60% of samples, are from the British Isles. So we might naively expect the entire haplogroup to originate in the British Isles, but 75% of the immediate downstream branches are Scandinavian. That's 6 out of 8 downstream branches are Scandinavian. Here you can see that better. So the haplogroup should be placed in Scandinavia, not the British Isles. Taking this tree structure into account is important to avoid sampling biases caused by both family size differences and database size imbalance as well. Okay, so we place the point in Sweden. And we do this process from every sample of the tree up to the root. So we've gone from the tips of the tree up to the very top. What now? Once finished, there are some steps that help us improve the results. For one, we snap any oceanic locations back to land. Hopefully most people didn't give birth at sea. Another improvement is called top-down smoothing. In this example, we have haplogroup RBY342 shown in purple. Its parent is in blue, and its 2 descendants in red are shown. Although this haplogroup and its ancestors have a long English history, one of 2 descendants is subsequently found in East Germany, which is a problem. That German coordinate would erroneously drag it from England east into the Netherlands. But we can use its English history as prior information about its likely true location. By comparing the distance from the grandparents to grandchildren, in other words, from blue points to red points, we can downweight the influence of outliers. So we're able to firmly place that purple point on English soil where it belongs. And this makes the map a little more conservative too, by smoothing out migrational zigzags that would otherwise occur. One last improvement is called TMRCA spacing. So this is where some of the age information is incorporated. This corrects for the fact that most of our sample locations are not ancient, right? They're modern testers. That would naturally bias Globetrekker to estimate less and less human movement as we approach modernity. And this is clearly wrong. To combat that bias, we space out the initial haplogroup coordinates according to their TMRCA. TMRCA spacing ensures that haplogroups are spaced out by the actual amount of time that they had to travel. And then it's important to note that any ancient DNA, of course, should carry a high weight and prevent much TMRCA spacing from occurring, right? If you have a sample that is 9,000 years old and it's on a branch that's 9,500 years old, then that should have a lot of weight. Once TMRCA spacing is complete, we have a spaced out path for every leaf in the tree. In this tree on the left, the red circle is an intermediate haplogroup, and it has 2 sons shown as B and C. These 2 paths give 2 independent estimates of where the red point should actually be located. Our last step is to average those together. We call this the mean path intersect, or MPI. This MPI represents our final estimate of the haplogroup's location, and the spatial uncertainty around each haplogroup is shown as what we call the hotspot. We show that to users. All right, now to the sort of interesting migrational paths that we all like to look at and stare at on the map. So, did all our ancestors move like Otzi the Iceman, scaling icy cliffs to reach their destinations in perfectly straight lines? Probably not. We think human beings generally chose routes that avoid terrain. This inspired the final innovation of our method, least cost paths and least cost corridors, or LCPs and LCCs. Now that haplogroups are pinpointed, these are the migrational lines that connect them. LCPs attempt to find the easiest route between 2 points, and by easiest, we currently mean the least steep terrain across land. If water is involved, then preferring coastal waters, waters that are closest to land. In rare oceanic travel, ocean currents, both speed and direction have an influence. So note that land here means whatever land was exposed during that era of the haplogroup. It could be, a lot more land at one time than another. LCCs are akin to confidence levels for LCPs. They show path uncertainty. It's pretty interesting to display these travel corridors, weaving in and out of canyons and cities. And a cool side note, I'm stealing this idea from my own published research on toads. So I'm stealing from myself, and this made the front cover of Heredity last year. So that's it. Here are some quick examples of the terminology. This is Native American haplogroup C, BY63635, and you can see the haplogroup point, all its descendant haplogroups together in the descendant shape. That just shows all of the downstream migrations together. And then there's the hotspot I mentioned. There's the corridor that I mentioned, and they both show uncertainty. Here's Polynesian haplogroup O, BY66844. Notice how Indonesia was partially landlocked back when sea levels were lower. It's one shelf. Here you see both upstream haplogroups and descendant shapes. Also notice the sibling lines for haplogroups that are siblings to the main ones. In one case, a sibling line connects to an interesting ancient sample out in Guam there. A couple more cool examples. Miguel's research has shown that haplogroup K spread quickly from west to east. Haplogroups K2 and P were already in Southeast Asia very, very early on. And eventually the ancestors of Q and the mega haplogroup R made it north of Lake Baikal, Russia, before branching 36,000 years ago back to the west into Europe and out eastward across Beringia into America. So very interesting that K was very, very early on in Southeast Asia. And this is evidenced by M, N, O, P, and S all arising in Southeast Asia, and by many indigenous groups still having those locations and those haplogroups. Another cool example, haplogroup EPF2546, which spread out from Carthage via North Africa. Hannibal famously led Carthage to victory against the Romans migrating from Tunisia through Spain. During that exact time, 220 BCE, we see several branches crossing Spain and Sicily. And this is presumably either Hannibal or one of the many Carthaginians ransacking early Rome and crossing the Alps with, war elephants and stuff like that. Very, very cool. All right, that's it. Remember to check and triple check your kit's EKA coordinate and country found under account settings. Globetrekker relies on the accuracy and consistency of user data, so thanks for helping us out as always. In the future, we may try to improve things by trying to add new environmental features such as lakes, encourage certain paths such as Roman roads in early Europe, and eventually we hope to integrate post-Columbian travel as well. It's a bit more complicated. To conclude, phylogenetics is not simple. Before this talk, you might have pictured this, a colorful map with a nice tree diagram. Now it may seem more like this. Sorry about that. But hopefully you have a better understanding, at least, of what's behind the curtain. It's really an exciting time to piece all these years-long projects together, and I couldn't be more proud of what our R&D team has accomplished with Discover. So with that, I'd be happy to chat with you and to answer your questions. Thank you.